Hey everybody, so today we are going to be talking about biosignaling. It's a topic that we actually haven't really covered too much on with our 15-week program that we were doing before. So now what we're trying to do is every week we're going to release some videos with some topics that we missed, and we're just going to go over them a little bit more in depth. So this week we're going to be talking about signaling, signal transduction, biosignaling, and a couple of related topics as well. My name is Chris. And just a general introduction for Socratic Med, who we are. Socratic Med, this comes from the website. Socratic Med is a grassroots nonprofit to provide sensible solutions to students with disparate medical school opportunities. So basically what that means for those of you that are not familiar with Socratic Med is that we try to help you get into medical school. So we post free content. I put some of the links down here. We have an Instagram, YouTube. You can go directly to our website, which is a great resource. We do one-on-one counseling, one-on-one tutoring, and we just finished our 15-week course. So like I just mentioned, we're actually releasing some more information just to fill in the gaps and just provide you guys with even more study materials. So on a more personal note, my name is Chris Scagnelli. I am a tutor here at Socratic Med. I graduated from Stony Brook University in May of 2019, and I was on a pre-medical track with a Bachelor of Science in Applied Math and Statistics. This past April of 2021, I took the MCAT and I scored a 520, which is the 97th percentile. And I did get a perfect score on the psychology sociology section. So my specialty in terms of tutoring is psychology, sociology, and a little bit of biology as well. So if you have any questions about pretty much anything I go over in this lecture, if you have a question about that or just any, any sort of content review, feel free to email me. I put my email down here and I'll also drop it again at the end of the lecture. Okay, so on to the actual content. So we're going to start the lecture just talking a little bit about signal transduction. At this point, you should be familiar with what it is, basically, what its function is, how it works. So I put this graphic here not to stress you out, not to, make it seem like there are 50,000 things that you have to remember. Just you have to keep in mind that when we're talking about signal transduction, we're talking about a bunch of things going on all at once. So, there are hundreds, potentially thousands of signals that are being transmitted through the cell at any given second. And a lot of times these signals have multiple targets. So it's very complex and it's very delicate. So that's basically what I wanted to, just summarize with this picture. But in no way do you need to remember these names, anything like that. You just have to understand the general flow. And we're going to talk about that right now. Starting at the top, you have those surface receptors. So what is signal transduction? Cells can communicate with the outside world via surface receptors. So when the extracellular domain of a surface receptor is activated, the receptor will effect a change inside the cell. So if we look right up here, these are two proteins. They're both most likely receptor proteins. And you could see this top over here where both of these proteins have these sort of two heads sticking out. That is the extracellular domain, because that is where the receptor is actually reaching outside of the cell to communicate with the extracellular environment. So that's, if we've spoken about receptors before, we talk about, induced fit model or the lock and key model. This is the lock part. So the key would actually be the ligand, or, that cell that's going to come and sit right on top of the receptor and start the signal cascade inside. But that specific part that the ligand binds to, we refer to as the extracellular domain, and that's the part that's actually sticking outside of the cell. So receptors may be activated by many different types of signaling molecules. These signaling molecules are also known as ligands, Ligand, as I was just referring to it before, same thing. But they can be hormones, neurotransmitters, they can be cytokines. It really depends on what the pathway is and what the target is. And some receptors don't even have an extracellular domain, and they're located deep within the cell. So for example, when we talk about the different types of hormones, one of them, these steroid hormones can diffuse through the cellular membrane. So a lot of times they have these receptors that are deep within the cell, like estrogen, for example. It works very deep inside the cell. It's not a surface receptor. But it still does, result in many of the same things as these do. I mean, there's a signal cascade that follows. there's signal amplification, and then it affects the entirety of the cell. So the job of a surface receptor is to transmit information from the outside of the cell to its internal environment without ever letting the signal molecule pass through the cell membrane. So in most of these surface receptors, nothing is actually passing through the cell. So the receptor will utilize second messengers inside the cell to carry out the signal cascade started by the original ligand. So that's why we have these secondary messengers. The ligand, if you look over here in this diagram, the ligand are these little orange pieces. That they're also referred to as the primary messenger because that is where the signal starts. So that is the primary messenger that is going to start the signal cascade. It interacts with the receptor, and then it does not pass through the receptor. The ligand stays on the outside, and when it's finished doing what it has to do, it'll probably just dissociate from the receptor and go back into the extracellular environment, where maybe it'll be degraded or taken up by something else, but it does not actually go into the cell. So because it doesn't go into the cell, that is why we have to utilize these secondary messengers. So those are the ones that actually carry the signal cascade. It's sort of like a relay of information from one molecule to the other. That's basically what the signal cascade is. And at the bottom of the screen, just to define signal cascade is the series of biochemical reactions that follow the activation of a surface receptor by its ligand. So the point of the signal cascade is to amplify the signal as it proceeds. So if we look at the diagram to the left and we go in order, we have 1, the receptor ligand binding. So that's going to start the signal. 2, signal transduction via the secondary messengers, whatever that secondary messenger may be. We'll talk a little bit about it in a couple of minutes. If it's a G protein, for example, then that receptor is going to mobilize that G protein and it's going to start its effect throughout the cell, which is 3, the cellular response. And then, oftentimes it is a change in gene expression, because when we're talking about this, and especially when we're talking about, cancer cells later, we're really talking about changes in gene expression that regulate cell proliferation and cell growth. So those are super important things to keep in check. But they're also super important things, to make happen as quickly as possible. So signal transduction allows us to do that. Okay. And we're going to talk a little bit now about cyclic AMP, also known as CAMP, and the enzyme known as adenylyl cyclase. So adenylyl cyclase is also known as adenylate cyclase. It's the same thing. It's an example that is very often used, especially when talking about signal transduction. And it will probably pop up on the MCAT on some sort of section. If it's not in the biology section, then maybe it'll pop up in the psych-soc section in something that you're reading. So it's a really good idea to be familiar with this. You definitely should understand it. So adenylyl cyclase is an enzyme which converts ATP into cyclic AMP, which is also known as CAMP. So when adenylyl cyclase is activated by its G protein coupled receptor, the second messenger CAMP then carries on the signal. So G protein coupled receptor is a type of surface receptor. If you look right over here at the diagram at the bottom, and we start all the way over to the left, this sort of oblong blue is the G protein coupled receptor. And then you can see 3 smaller purple things attached to the bottom right. Those are the 3 subunits of the singular G protein. So the G protein is broken down into alpha, beta, and gamma. All of them have their own rules. But the important thing to know is that when that G protein coupled receptor is activated by its ligand, that's going to activate the G protein. So the G protein is going to dissociate. It's going to exchange GDP for GTP. It's going to become active. And then it's going to activate adenylyl cyclase. And then adenylyl cyclase is going to turn ATP into CAMP, cyclic AMP. So you could see how it's very sequential. And then once cyclic AMP is made, the levels of CAMP in the cell are going to rise. It's going to activate protein kinase A, and it's just going to continue the signal cascade. So it's like just a very sequential, step-by-step process when we're talking it out like this. But like I mentioned before, keep in mind that there, thousands in a given second are going on, and your cell is doing all different kinds at all different times. So there are a bunch of weird interactions that, in vivo actually do happen. So right, pKa, which is protein kinase A, will then phosphorylate and affect the other target cells, and it's going to contribute to the metabolism of glycogen in the cell. So pKa is actually activated when we're trying to mobilize our like glycogen stores or our fat stores for like a fight or flight response. Okay, so I'm going to put a review question in here for you guys. I would take maybe no more than a minute to answer this. I'm not going to wait a minute, so just pause, take your time, and when you're done, you can unpause. But which of the following is not true regarding signal cascades? So just read the 4 answers, and then we'll see what you have. Okay. So which of the following is not true regarding signal cascades? So the answer to this question is going to be D, and let's go over it. So A, the signal originating from one receptor will be amplified many times over as it travels down its pathway. So the question is asking which of the following is not true, and that is 100% true. That is the whole point of the signal cascade, is that it's amplifying the message. So it's a really effective way to transmit information throughout the cell. So A is correct. A is a true statement, excuse me, so it is not the correct answer. Because remember, again, we're looking for what's not true. So B, some signals may be first received by receptors deep within the cell. Well, we did speak a little bit ago about how estrogen works like that. It goes deep into the cell because it's a steroid hormone, so it can actually pass through the cell membrane just by simple diffusion. So some signals do actually start with receptors deep inside the cell. So B is also a true statement, so it is not the correct answer. C, cyclic AMP, also known as CAMP, is a common second messenger in humans, which it definitely is. We just talked about the CAMP pathway. Your cell uses CAMP levels to adjust pKa activity. So like we said, if CAMP is upregulated, your cell is going to upregulate pKa, and we're going to mobilize those glycogen and those fat storages for the fight or flight response that we have. So C is a true statement, which makes it not correct for this question. And then we finally get to our correct answer choice D, the strength of the signal diminishes as it is transduced throughout the cell. That's not true. It's actually directly opposite of what A is. So if you knew that one of them was, if you could identify that they're sort of antonyms, then you would be able to narrow it down to just A or D. And hopefully you would remember that signal amplification is a part of signal transduction. So the signal actually gets stronger. So D is not true. So that makes D the correct answer. Okay, moving on to our next topic. We're going to talk a little bit about the 3 types of surface receptors, and we have them up there. So we have G protein coupled receptors, catalytic receptors. I believe this is a receptor tyrosine kinase in the middle over here. And then we have ligand-gated ion channels, which we're going to talk about. So a little bit more about surface receptors. Cell surface receptors are usually transmembrane proteins, and they have 3 domains. So transmembrane, remember, just means that it's sticking on both sides of, it's sticking through the cellular membrane. And domains are just different parts. They're just different functional parts of each protein that have, different functions. So we have the extracellular domain that we spoke about before, which is also known as the recognition domain. And it's exposed on the outside of the cell, and this is what binds to the ligand. So this is what starts the signal cascade. It's sticking outside of the cell, hence the name extracellular. And it recognizes the specific ligand for that receptor. So that's also why it's known as the recognition domain. So the membrane spanning region is made up of hydrophobic proteins, and it spans the cell membrane, is exactly what it does. So that's also a pretty self-explanatory name. And if you recall that the cell membrane is hydrophobic because it's a phospholipid bilayer, then that should be pretty intuitive to you that these hydrophobic proteins are the ones that are actually going to stay lodged in that membrane. So the makeups of each domain are a little bit different, and you could see that it sort of interacts, with the physiology and with the physical properties of the environment. So form does match function here. So the intracellular domain is the 3rd one, and it's also known as the coupling or the catalytic domain, and it's the active site, and it's located just inside the cell membrane. So we say active site as just a general, just sort of like a general term that some sort of catalytic activity is going on in there, but it varies greatly depending on what receptor you're actually activating. which we're going to talk about right now. So the first and most familiar one is probably the ligand-gated ion channels. So these are gated channels that swing open when the ligand is present, allowing for the passive flow of ions. So if you look to the right, this diagram, you have this these are, we can look at these as the same channel, the same receptor. So if you look to the left, it's in a closed state. There are ions that are outside. Clearly, they're higher concentration on the outside of the cell than they are the inside. And it looks like the ligand for this specific ion channel is some sort of neurotransmitter. So that neurotransmitter is going to bind to the ligand-gated ion channel, and then it's going to switch open, right, like it is to the right over here. So when it switches open, it's a channel. So unlike the G protein coupled receptor that we were talking about before, there is an actual connection. There's a communication between the extracellular environment and the inside of the cell. So that's what a channel is. It just allows these ions to flow down their natural gradient. So it's not pumping, it's not using ATP to like pump ions in or out of the cell, but it is a channel and it is allowing them to flow passively. But it can only allow these ions to flow if that neurotransmitter or whatever other signaling molecule attaches. So that's the most important part about ligand-gated ion channels, is that you can have a huge concentration gradient, but as long as this gate is not closed, those ions are not going to go through the cell membrane. So quick changes in ion concentration are very important in nervous and muscular functioning, as I'm sure you guys are all familiar with. And the most common ions that we see in these channels are usually sodium, calcium, and sometimes hydrogen ions and magnesium ions as well. And these receptors are different than the other two in that the transmembrane domain creates a physical channel that connects the cell and the extracellular environment, like we said before. And there are other types of ion channels, like voltage-dependent ion channels. But in terms of today's lesson, biosignaling, ligand-gated ion channels are definitely the most pertinent to this topic. So we touched a little bit upon G protein coupled receptors before, but again, GPCRs are the largest type of receptor. Upon their activation by an extracellular signal, the GPCR activates a trimeric GTP binding protein, which is also known as G protein. So we spoke about this a little bit before, but looking at this diagram to the left, this ligand is going to attach on the extracellular side. This is that binding domain that we were talking about before, the recognition domain. So when it does bind, this G protein, which is right over here, it was purple in the last picture, and it's actually green and purple in this picture, but it still is that trimeric alpha, beta, and gamma subunits. That whole protein is going to dissociate from the actual receptor whenever there's a ligand present. And then that GDP is going to be switched for a GTP, and then the G protein is activated, and it can go on and act as a second messenger, and it can affect whatever change it is going to affect inside the cell. And it just continues that signal cascade. So like we said, when the G protein exchanges a GDP, for GTP, it switches to its activated state. When the GPCR, that should say, is activated by its ligand, its G protein acts as a second messenger. It can open ion channels or act enzymatically on a target in the cell. So it has a couple of different roles, a couple of different ways that it can, affect change inside the cell. But this is a very important receptor. I think it makes up like half of all receptors that we target, like pharmacologically, in terms of treating disease. It's implicated a lot in pharmacology, and it's a very hotly studied field right now. Okay, so we also have a class of receptors called catalytic receptors. They're also known as enzymatic or enzyme-linked receptors. So these receptors have an active site on the intracellular domain where the catalytic activity may occur. So they're a little bit different than G protein coupled receptors, where if we go back and we look, this G protein coupled receptor, once it is activated, it allows this G protein to dissociate from the receptor, and they are now two different pieces. And that G protein is what travels throughout the cell and is the actual catalytic component of the GPCR. In terms of catalytic receptors, these are the catalytic activity actually occurs on the receptor, but it's an intracellular domain. So they're often kinases. So whenever we learn about catalytic receptors, I know in like cancer biology classes, when you take them, you'll definitely learn about receptor tyrosine kinases, which I think are one of the more common catalytic receptors, and probably one of the ones that you'll see the most. So I believe this, yes, so this diagram that I have here to the right actually is a diagram of a receptor tyrosine kinase, which is just a more specific family of catalytic receptors. Yeah, they do what catalytic receptors do. So they phosphorylate other molecules. They actually phosphorylate themselves in a process called autophosphorylation first, and then that autophosphorylation is what activates the actual catalytic domain of the receptor tyrosine kinase. So usually it phosphorylates itself, and then the RTK will phosphorylate another molecule inside the cell, and then that molecule acts as the receptor tyrosine kinase's second messenger. So again, when we looked at the GPCR, the second messenger for the GPCR is the actual G protein. But if we have a catalytic receptor, it's going to phosphorylate some other molecule that's been in the cell, and that phosphorylated molecule is what is activated, and that is what's going to act as the second messenger, and it's going to continue the signal cascade. Which you could see when you look at the diagram over here. They're sort of, it's two separate receptor tyrosine kinases are actually sort of two separate proteins. And then when they are phosphorylated, they dimerize into one protein. And that's actually how they activate. So like the actual physical conformational change of the receptor tyrosine kinase is what activates its catalytic activity. And it allows for all this entire signal transduction. Okay, so we're going to talk very briefly about tumor suppression and apoptosis. So that's just, a quick picture, normal cells and cancer cells. You can see cancer, for those who wouldn't know, cancer is just an unchecked growth of normal cells. A bunch of different ways how cells can go haywire. We're not going to focus on that too much in this lecture, but it is important for this discussion. So tumors are often caused by mutations in genes that regulate cell signaling. Cell proliferation is regulated very delicately by each cell. Like we said in the beginning of the lecture, with that huge picture with all of those different, signal pathways. But a disruption in any of those signal pathways can result in unchecked and potentially malignant tissue growth. And some receptors may even fire independently of extracellular ligand binding. So, it's usually mutations, or, we could talk for hours about how cancer starts, the different pathways, the checklist of things a cell has to go through. But receptor tyrosine kinases are one of the types of receptors that are very often implicated in cancer. And I just put another graphic at the bottom so you can take a look at that. The difference between ligand-dependent firing and ligand-independent firing is right here. So the one on the left right over here is only going to fire, and it's only going to send out, proliferative signals to the rest of the cell if there's that GF growth factor is attached to this receptor tyrosine kinase. Sometimes when these receptors are mutated and the cell starts pumping out a bunch of them, you could see over here, one of a couple of things can happen. It could either stay close, or there's no actual extracellular domain on it. So there's no extracellular domain. So these are just dimerized constitutively, and they're just constantly sending out these signals, which is an issue because, again, unchecked cell growth leads to tumors. It could lead to metastasis, and it could lead to death. So your cells have developed a way to monitor cell growth and destroy any cells that are proliferating too fast. And then they're in a bunch of different ways. They have just like natural cell checklists. Your immune system plays a role as well. But what we're going to talk about today, one of the more important proteins that your body makes, is known as P53, and that is sort of the famous human tumor suppressor protein. So the gene that encodes for P53 is a gene that specifically encodes this protein to check tumors, to keep tumors and unregulated cell growth in check. So p53 protein levels in a damaged cell will rise and eventually trigger a signal cascade. So if we look to the right over here, if you look at the p53 response at the bottom, whenever there's like an increase in p53 production, that obviously means that there's a problem with the cell. So it will try one of those, one of those 5 things. The cell could enter senescence. Cell cycle could be arrested until maybe the cell tries to do DNA repair or it tries to figure out what's going on. The cell could also differentiate, but a lot of times when there's nothing left for the cell to do, when it's not contributing, and it could even, and the body recognizes that it could be a threat to the surrounding tissue, it undergoes apoptosis. And that is, again, a very popular signal cascade that you may see on the MCAT, which we're going to talk about in a couple of minutes as well. We'll talk about right now, actually. So the apoptotic signal cascade, apoptosis is the pathway for programmed cell death. And it is a last resort strategy, which destroys the cell from the inside out so as not to damage any surrounding cells. And that's super important because there are a bunch of different ways in which cells can die. Necrosis, autophagy, there are a bunch of different ways they can die, but apoptosis is specifically programmed for cell death. So it's just the body's way of getting rid of cells that we don't need anymore, and it does it in a very safe way so that it doesn't damage any other healthy cells that are around it. So it's sort of a very quiet fizzle out rather than a huge burst, as like a cell that is lysing would be. But during apoptosis, the cell first begins to shrink. As the cell shrinks, the cytoskeleton is disassembled. The cell starts to bleb, is what they call it, and it starts to get sort of bubbly and it starts to bud out because the cytoskeleton isn't really holding anything together anymore. So the nuclear membrane starts to break down, the genome starts to become fragmented, and then the tiny remains are broken down and recycled by phagocytic cells. So if we look at the bottom, exactly what I just said, there's a normal cell, the cell shrinks, the chromatin starts to condense. You could see over here, the membrane starts blebbing, the organelles start to disintegrate. There are caspases, which we're going to talk about in a second, that are just flying around the cell. They're destroying the proteins. Everything's being broken down. The cytoplasm is blebbing off, and it's just breaking down into tiny little remnants of the cell that it used to be. So then these tiny little apoptotic bodies form, which they're basically just little pouches of broken up protein. And then they're digested, and they're usually, if there's anything that's recyclable or reusable, the cells will do so. But that's basically what apoptosis is. And as far as the actual signal cascade, apoptosis is carried out by that cascade, which involves enzymes that are known as caspases. So caspases are a class of proteases. Remember, proteases cleave proteins. And the reason why they're called caspase is because they use a cysteine to cleave target proteins at an aspartic acid residue. So you can see that C comes from cysteine, the ASP comes from aspartic acid. So that's where the name caspase comes from. And there are actually 2 types of caspases. So the first one that we'll talk about are called initiator caspases, and obviously the 2 of these have 2 different roles. The initiator caspases are the ones that are first activated by whatever the first death signal is. So for example, the example that we've been using is p53. Whenever the cell sees that there's stuff going on that shouldn't be going on, maybe that cell's becoming a little bit malignant, maybe the p53 levels are going to increase, and that signal transduction is going to take place, and that's going to result in the activation of these initiator caspases. So these initiator caspases are going around, and they are going to activate more initiator caspases. So they activate themselves, and they also activate effector caspases. So that's also how the signal is being amplified. If you initiate one initiator caspase, for example, it's not just going to be one initiator caspase starting all of these effectors. It's going to start initiating other initiators as well. So that is where the signal amplitude comes in. So then it just grows, and it's a sort of snowball effect. And the effector caspases that are activated circulate the cell, and they break down proteins to trigger apoptosis, like we just mentioned before. Okay, so here's the final review question. So which of the following statements is or are true about caspase cascades? Take a minute, read the 4 of those, and I'm going to go over them. Okay, so which of the following statements is true about caspase cascades? So the answer to this question is C. And we could read all four of them and we'll just go through them individually. So Roman numeral 1 says, initiator caspases activate effector caspases. So the question, remember, is asking which of the statements is true. So initiator caspases do activate effector caspases. So that is a true statement. Roman numeral number 2, initiator caspases activate other initiator caspases, which is also true. So since we know that 1 and 2 are correct, we can actually automatically eliminate A and B because 2 is not a part of their answer. So when we look at Roman numeral number 3, again up here, the effector caspases activate initiator caspases. That's not true. The effector caspases are the ones that are actually carrying out the catalytic effects of apoptosis. So they're actually going around and they're cleaving all of these proteins. The initiator caspases activate effector caspases, but it does not happen the other way around. So 3 is not correct. And then from there, you can eliminate D, and that leaves us with C. And obviously 4 is correct as well. Caspases cleave proteins at the aspartic acid residue. So that's true as well. And happy studying. That's all of the content that I have for biosignaling, but be sure to check back. We're going to be uploading more videos. I think we're doing about 2 every week. And if you haven't already looked at our 15-week course that we finished a couple of months ago, you definitely should give it a look. There's a lot of great content on there. Like I said before, you can look at it on our website. You can get it from our YouTube page. And again, if you have any questions about anything that we went over today, or any questions in general about being pre-med or about the MCAT, please don't hesitate. Shoot me an email over there. I may not be the quickest at responding, but I will try my best to get to everyone. All right, everybody. I hope it was helpful. Until we meet again.